So the hypoblast is the primitive endoderm, and the epiblast is the primitive ectoderm. And the amnion forms to eventually surround the fetus. So for us, I mean, frogs, give birth to eggs. I mean, so we carry that a little bit farther. So you have to kind of understand, he said that all animals go through this process, but a little bit different stages. So for us, we don't get that. Mesodermal development does not happen until after this stage. And see, gastrulation is the process in which a gastrula develops from a blastula by the inward migration of cells by a complex and coordinated series of cellular movements which occur at the end of the cleavage state of development. And then eventually we're going to end up in this. So first we have a bilayer disc, and then we will eventually end up into a 3-layer disc. And notice those words, hypoblast, and I got the memory down here, because sometimes I think the vocabulary is really secret. So hypoblast is really the endoderm. And the epiblast is the ectoderm. Now, we could probably actually get that from just the words epi and hypo. So epi is around and hypo is below. If you ever kind of have a hard time remembering hypo from hyper, just remember hyper is someone who is hyperactive, they're really hyperactive. So hypo is the opposite. So the hypoblast is the endoderm, and the epiblast is the ectoderm. And then now we're beginning to see, you're going to see the yolk sac form into the former blastocyst cavity. So there was a blastocyst cavity. Remember that was, that disappeared in the frog egg. And so this is the beginning of the yolk sac. And then we have this bilayer disc positioned between the amnion and the yolk sac. And this gives the primordial germ cells. And then this extra embryonic mesoderm begins to develop from the yolk sac. And then these cavities then form this extra embryonic coelom. And this would be this cavity, a coelom, like he said, blastocele is a cavity, so the coelom is this cavity. And you can see, so for us, the mesoderm develops a little bit later, but of course, and why is any of this important? Well, it's just important to understand this development and how, like, to understand our body. You could still think of our body as this 3-layer disc, really. We're a tube within a tube, but we're of this 3 layers in a very simplistic form. And what you see a lot of pictures like this of this is this gastrulation. And I actually have a little video you can see, if you want to see it, we can watch it, it's only a couple of minutes long, about the development of this primitive streak. And so for us, for humans, we develop this primitive streak. The cells migrate down into this streak, and then that eventually, like he said, will become part of the notochord and the somites and all of this development. The 2 layers become 3. So this is the development of the mesoderm, and then this development of this notochord process through, so the cells migrate down this primitive streak. And now eventually the yolk sac disappears, and the amniotic cavity is the fluid that surrounds a fetus in utero. You guys are looking at me like, I'm really confused. And then of course we have this further folding. Neurulation would be this folding of the spinal cord, and we even have this folding into our brain. You can see here is this neural tube developing, and the red is the mesoderm. So the colors. One nice thing about anything embryonically, the colors always relate to these layers, and they don't change. So you're always going to see the, I can't read my slide either. Anyway, this is kind of hard to read. Can you guys even read that from here? No. Not very well. Let me see if I can get it here. So here is the endoderm. Here, this inner line. This would be the mesoderm, and then the ectoderm. And these are the cell-mite development, the cross section through the embryo like this. I don't know why this.
Yeah, I think it's all out of the book.
Pardon?
These are slides are out of the book, right?
The slide is what?
Out of the book.
It is out, yes,
Yeah,
this is in the
it's
book.
in the book.
Yeah, all of this is in the book, so you can get it from the book. Yeah, we really, I mean, you should be able to see these more clearly. Anyway, here again, you can just see this, this is just this more and more of this folding. And you can see here is, the gut beginning to form, and and you can begin to see that this does even kind of begin to look like that. The early stage, you can almost begin to see the vertebral bodies in there. And then here are just some actual pictures of 20-day embryo, the neural plate, the neural groove, and the amnion somites bulk sac. So this neural crest, one of the things that we'll talk about skeletally, and you guys have had the skull lab this week, and that skull bones develop very differently from the rest of the bones of the body. And I think a lot of this comes from this neural crest. At least when I talk to other scientists, they go, oh, it's the neural crest. Always the neural crest that causes this big variety that variation, it's not really a variety, but variation that we have from our skull to the postcranial. I mean, our skull bones form differently. Our facial muscles they're muscle tissue. They're just like the rest of the body, but they have a little bit different action. I mean, they work on skin, not on bones. They aren't moving bones around, they're moving skin around. Not all the facial muscles occur, they don't all attach bone to bone. Many of them don't ever even touch a bone. They're just located within the skin itself. And there was a popular television show a few years ago called Don't Lie to Me, or Lie to Me, or something like that. Anyway, it was really great. It was about a guy who was at UCSF, did a lot of research on facial expressions. And one of the things they discovered is we have these micro expressions. So we think a thought and it appears on our face. And even actors or someone who has really trained themselves to have no reaction, poker players or whatever, people have trained their faces to have no reaction, still have this micro expression. And it's because our facial muscles and our brain and all of this, I think, is also interrelated. Of course, our whole body is interrelated, but I think there's a lot of special relationships between the skull and the brain and the face. And of course, that's a major communication area for us too. So here is just another 24-day embryo, the developing brain, the heart, and again, these somites. And the somites, we talk about humans being segmented animals, and we really are segmented. I mean, all you have to do is look at the vertebrae. I mean, we're in segments. I mean, we don't think of ourselves as segmented, but we are. We really are segmented animals. Our nervous system works in segments along the vertebra. And these somites form in segments. You can see this kind of body plan from day one. And here is a little bit older embryo. So this is an embryo, not a fetus. And to me, there's a very distinct difference between those two stages of development. But this would be an embryo, and you can see the beginning limb buds and the pharyngeal arches, gill arches. I mean, if you took a fish and you put a fish and a human next to each other at certain stages of embryonic development, you might not really be even able to tell the difference. And in fact, some of our skeleton we call the brachial skeleton because it forms from these gill arches. Mandible, the inner ear bones, the hyoid bone, and I think the inner third of the clavicle, something like that, is part of this brachial skeleton, which is part of these gill arches that for us have developed into a little bit different structure. And then, I don't know if I talked to you about this, but the whole, something a colleague of mine and I discovered, we didn't discover it, but we were talking to each other and we suddenly realized that humans, we're still locked into that aqueous environment in terms of our breathing. Even though we're breathing air, the air is dissolving into the plasma before it goes into our red blood cells. So it's still an aqueous environment for us. Anyway, and here you can see 44-day embryo developing soon into the fetus. You can see the umbilical cord and the placenta, and all of that, all the placenta, all of these things develop from the developing egg. The uterus doesn't provide those. The fetus provides that as part of their development. We can go backwards a little bit. Let me show you. So you can see, here we go. You can see that this is the beginning of the placenta here, which is, so this is, this all comes from the embryo. So here would be, and this is a picture of a fetus. So this would be the 10-week-old fetus. And you can see that now all of that formation, now we have this, placenta, the umbilical cord, there's the yolk sac, soon to disappear. And you can begin to see that, we can recognize this as human. And then 13 weeks, and then 26 weeks. Okay, so you guys want to see that film again? Anybody want to see it again? No? Because that's embryology in terms of anatomy. Like I told you, I'm not going to spend a lot of time on this, but I do want you to understand it. And I do want you to download this. Does everyone understand what the archenteron is? The primitive gut is this archenteron. And this is formed by the central cavity of the gastrula. The inward migration of those endodermal cells, which eventually become the lining epithelia, the epithelial lining of the digestive cavity in the gastrocoel. So you can see kind of how all this is, hopefully you can see how it's all kind of, we're building these blocks to eventually make a human. Any questions? Yeah.
His name was George Wolfe, you said?
Oh, his name is George Wolfe.
Okay.
Yeah. And a student told me about that site, this Mindwell, or what, Mind Bites or Mindwell. Anyway, they told me about that, and they're not expensive. I'm not selling them or anything or telling you have to buy them, but he told me he got through, I don't remember what it was, some chemistry class using this site. So I think, you have to dig through the internet. I mean, there's a lot of things out there that are no good at all, or that are kind of interesting, but they're not really informative. But there are a few things that are really, really good, and when I find them, I'll try and bring them to you. If you want to watch that again, I can show it to you again, but if you feel like you understand it, we'll just move on.
Can you just put a link of that on iLearn?
I embedded it in the PowerPoint. Has anyone downloaded that PowerPoint? I think it'll come in through the PowerPoint. If not, then I don't think since, I don't know, you have to, I think I have to pay $3. I mean, not paying a lot of money, but I don't know that I can just put it up there. But I did embed it in the PowerPoint, so it might show up if you want it. And I just wanted to give you his name. They never give his name before, but I look it up, because he does a good job, and I want to give him credit for it. Anything else anyone wants to talk about? The amniotic cavity is an amniotic-filled fluid cavity surrounded by the developing embryo. We know that ectoderm. Well, let me read to you what is formed from the ectoderm. So the ectoderm, the outermost layer of the 3 primary germ layers of an embryo, which develop during the transition from blastocele to gastrula stage, from which the epidermis and the epidermal derivatives, like hair, nails, tooth enamel, sweat, sebaceous glands, and the nervous systems and its sensory receptor cells, such as the retina, the spiral organ, gustatory cells, etc., develop. So anything that is on the outside of our body has come from this ectoderm. The endoderm, the innermost lining, the innermost of the three primary germ layers of an animal embryo which developed during the transition from blastocele to gastrula, developing into the epithelial linings of the gastrointestinal tract, the lungs, associated structures, and all digestive glands, and most endocrine glands as well. So that's kind of interesting. And then the mesoderm, the middle embryonic germ layer lying between the ectoderm and the endoderm, which developed during the transition from blastocele to gastrula stage, from which the connective tissues proper, such as bone, cartilage, dentin, which is your teeth, cementum, which surrounds the tooth, all types of muscle, blood, endothelium of blood vessels, synovial membranes, serous membranes lining the body cavities, the epithelium of the kidneys and urinary drainage system, and the germinal epithelium of the gonads and linings of the reproductive tracts. So, clearly the majority of our body comes from mesoderm. And this is online. You can just get it from online.
So like all the visceral layers are ectoderm, or endoderm?
The linings of, visceral layers. That's a really good question, because technically the visceral layer is on the outside of the organ. Even though it's the visceral layer, that's a really good question. And this is the kind of things that where your mind can get kind of twisted in half. Yeah, okay, so when we think about, during this gastrulation process, we just saw it in this little 3-layer germ layer, but you just have to know that went on until the whole gut was developed. As it folds through that tissue, like the balloon, there is a tissue that touches the organ, and then there is an outer tissue. I believe those would both be ectoderm. Well, not ectoderm, those would both be mesoderm. Those would both be mesoderm. So the inside, like the inside of the heart, would be endoderm. The inside of the digestive tube would be endoderm. What was the other one you were asking about?
But like the outside of the heart would be?
I believe, now that is a really good question, and I'm gonna look that up, because, I actually don't know, but that's a great question, so I'm gonna look that up, I'll get back to you. So the heart, I believe it would be technically mesoderm, but I don't know for sure. And that
Pericardium mesoderm also?
Pardon?
The pericardium also mesoderm?
I believe that would also be mesoderm. See, because it's actually, the endoderm would have formed the inner structures of the heart, like the chordae tendineae and all the muscle layers on the inside of the heart. The endocardium. So when you look at the heart wall itself, that's a really good question. And these are kind of things that, a lot of times, and especially for a beginning anatomy class, probably a little advanced for us, but, like on the heart itself, and this is actually, I'm going to do this, even though it's kind of a little bit of away from what we're talking about, just because it goes to language, which was what we were all kind of supposed to be working on this last 2 weeks. So if when we look at the heart, we have this pericardial cavity, and then we have, so the heart itself, so we have, so we would have, the heart has these 3 layers. It has a pericardium. It has a myocardium. myocardium, and it has an endocardium. Now the pericardium is here, the myocardium is this muscle wall here, and the endocardium is the layer on the inside of the heart. And my guess, and I don't know for sure, and I will look this up, is that these are both mesodermal and this one is endodermal, I believe. Does that make sense? Even though in the heart, technically, this pericardial layer is the visceral layer, and this layer, the pericardium, this layer that is surrounding the heart, and there's fluid in there, that this layer would be what, like when you think of that balloon, would be that parietal layer. We call it a little bit, we call it the pericardial sac, or the pericardial pleura. But anyway, yeah, that's a really good question. So, and we find this in our body a lot, these two terms, you know, pericardium and myocardium, endocardium, you know, around to the side, and myo is a muscle, and the endocardium is just this lining on the very inside layer. So, yeah, it's very interesting. I try to learn while you're learning, so if you don't mind. Now I do have, we can